So I'm here with Jeff Settleman, Senior Director of Discovery Oncology at Genentech, who's presenting work on the rational selection of therapeutics in patients here at the NCRI Symposium. Jeff, tell us a bit about why you're excited about this type of technology.
Sure. Well, I think we're really starting to see the power of the use of predictive biomarkers clinically in stratifying patients for treatment with some of these newly developed so-called rationally targeted cancer therapies. They're looking quite promising, particularly as we match patients with treatments based on molecular tumor features.
So the class of therapeutics you've been mainly focusing on have been inhibitors of tyrosine kinase receptors. And some of the exciting things you showed us was about that you can get dramatic retreatment effects in patients who've previously relapsed on primary treatment with cetuximab, for instance. Tell us what the implications might be for clinical practice in the future.
Well, retreatment responses, which so far have largely been seen in a limited setting because it's not standard protocol to retreat patients with the same therapies again. But these seem to reflect some newly appreciated reversible state of drug resistance that can arise during therapy, probably reflecting some epigenetic regulation, we believe, in tumor cells. And this creates some potentially new opportunities to target states of chromatin that might be conferring resistance in those settings. And we find that quite exciting.
So much of the work you showed was looking at cell lines and the evolution of resistance mutations. How might that be applied in the context of personalized medicine for individual patients?
Well, as we understand the full spectrum of mutational mechanisms to drive resistance, a couple of themes are beginning to emerge. First of all, the tumor really wants to maintain signaling function through the same elements that are being driven by the activated oncogene that it had become dependent on. So some of the key survival nodes downstream of a lot of the kinases, such as MEK and AKT, kinase, etc., seem to be playing a somewhat of a universal role in tumors. And so resistance mechanisms often involve keeping those pathways engaged. So if we understand how tumor cells achieve that, we can begin to develop rationally based second generation therapies to overcome resistance.
So one of the key nodes downstream is KRAS, and mutations are becoming increasingly recognized as an intractable form of resistance. What do you think we can do for that particular problem?
Well, KRAS, of course, is one of the most important oncogenes in cancer, and the percentage of tumors that harbor activating RAS mutations. Its role, as you mentioned, during the acquisition of drug resistance is also beginning to be appreciated. And yet, unfortunately, RAS as a protein looks like a somewhat intractable drug target so far. It's just the nature of the enzyme function of RAS and structure are not really amenable to good strategies for therapeutic targeting. However, the good news is that our understanding about the multitude of downstream effectors of RAS is such that a couple of those, particularly MEK and PI3 kinase, seem to be most relevant in many tumor settings, and we do have agents that target those downstream elements. And so, at least based on preclinical data, one can combine inhibition of MEK and PI3 kinase and perhaps achieve benefit in RAS-driven cancers, and that preclinical data is now driving clinical trials to test exactly that hypothesis.
Some of the work that you showed was essentially using sequential use of different agents targeted against the same target. Do you envisage that's the direction of travel, or should we be combining several targeted therapeutics simultaneously?
I personally favor combinations up front, but what limits that is toxicity, of course. As we start to combine things, we also combine toxicities, and this is really creating a significant limitation to what we can do up front with therapy. And just the nature of clinical development and the way trials are structured to combine multiple agents, particularly new agents, is challenging and takes time. But from a scientific point of view, we would like to take the approach that was so successful with HIV treatment, which is the so-called cocktail therapy, where we combine upfront, specifically with the goal of preventing the development of acquired resistance.
A long-standing criticism of cells grown in culture is that they don't reflect the natural situation, and yet much of the work you've presented actually says that they do reflect the natural situation for years after they were established. Do you think we've reached the zenith of the models that we're using, or are there other things we should be doing in parallel?
Yeah. Well, as you know, there certainly are caveats around the use of cell lines that have been around for a long time, maintaining culture. We know to some degree that they capture the kinds of genotype response relationships that we're seeing play out clinically, which is good news. I think a priori we would not have known that would be the case. Many would have predicted that it wouldn't be true. Fortunately, that has been demonstrated in some cell model systems in culture, but there are limitations. I think we're certainly not capturing everything. We know that there are very important elements of tumor biology in vivo, such as the contribution of the vasculature, of other stromal components that impact the response to treatment. That is either the ability to deliver drugs effectively to tumor cells, or the contribution of stromal produced factors that impact the properties of tumor cells with respect to their response to therapy. And in addition, the three-dimensional nature of tissue is obviously a difference that we can't capture easily in culture. And so there will be differences. The good news is we can capture some things that are certainly clinically relevant. We're certainly missing other things.
This may be difficult to answer as somebody working for one of the world's leading pharmaceutical and biotechnology companies, but what are the implications for what is now being discovered for the cost-benefit ratios for the new generation of therapeutics?
Yeah, well, certainly we've seen the model for drug discovery and development change rapidly as we've begun to appreciate the importance of patient stratification and personalized medicine and oncology. As you reduce the market potential for any drug, obviously that plays out in the cost-benefit analysis for developing any particular therapy. And by personalizing treatment, we are shrinking the relevant patient population that we're treating. On the other hand, we have examples where what was thought to be a significant limitation associated with that kind of market reduction turned out to not be as substantial as we thought. We have being a good example in the context of a disease like CML, relatively low incidence. But the reality there is that patients stay on therapy for a long time, and Lebeck found applications in other settings as well, and so you ended up with a blockbuster drug. So I think while we certainly can't develop custom therapies for every individual patient, I think that we can develop cost-effective therapies for a relatively small fraction of patients. What that number is, I don't know. That needs to be determined on a case-by-case basis, undoubtedly. But we're definitely revisiting the paradigm for how we develop cancer drugs. We don't just make drugs for each disease. We're really thinking about each patient.
Jeff, it's been a real pleasure talking to you, and a very exciting way to start the day at the conference. Thank you for your time.
Thank you.